There we go. Thank you for joining us today, Paul.
All right, so my name is Paul Vorster. I am a lecturer in the bioengineering department. And today we're going to talk about DIY biology or do-it-yourself biology. Where is my screen? So there we go. So what is on the menu for today? So brief introduction of who I am. What is DIY biology? We'll talk about the origins of DIY bio. And then we'll talk about, sort of as a comparison, we'll talk about big bio or more of a traditional lab. And I excuse me, I'll use the example of the teaching lab that many of our iGEM students have been in. We'll have a discussion about biosafety levels, and I'm gonna make sure nobody falls asleep during this part of the discussion because it is actually kind of important. And then we'll talk about something called engineered controls, how labs can be built in order to be safe. And then we'll come back and then just sort of compare DIY bio with traditional labs. And then we'll have sort of an open discussion about biosafety. And this part of the discussion will be led by the iGEM team since this is pre-recorded. So very briefly, who am I? Lecturer in the bioengineering department. I started working in research labs in 2004, graduated, started working in research lab in 2004. I worked in a drugs of abuse lab where we looked at the genetics of addiction. Very fascinating stuff. I was trained as a microarray lab tech back in the day, so I have a lot of lab work experience. After that, I worked in an HIV lab where I made viruses. I worked with the HIV live virus and I made other lentiviruses. And then I got interested in developmental biology and sort of the genetics of development. And so then I started working with zebrafish and frogs. I came to Stanford in about 2015, where I took a position as a postdoc and worked with fruit flies and then also worked with worms. So a lot of things. And while I did all this stuff, I was also teaching while I worked in labs. And I just really very much enjoyed teaching. So now I get to teach and share all of my experience with students in a teaching lab, which I really very much enjoy. All right. All right, so let me start with a question, okay? Are bacteria dangerous to humans? And I'm gonna ask the iGEM team to jump in and answer some of the questions just to help me with my presentation. So are bacteria dangerous to humans? Who wants to take a stab at this?
Well, not inherently, because we have like bacteria inside of us and on our skin and stuff, but some bacteria can be damaging in like different levels, right?
Okay.
Like 3% of our body weight, bacteria.
Yeah, that's right. Did you read my slides ahead of time? No.
I know.
Okay. So good answers, really, really good answers. I think a general misconception is that all bacteria are just dangerous. And you know how some parents would like excessively clean their children's hands and like spray everything with like antibacterial stuff and whatever. I would say in general, that's not a great practice, and we'll get into that in a little bit. But because of the public health crisis we're in right now, please wash your hands excessively. It's, do that right now. But in general, I would say there's a misconception where people think that, oh, bacteria is just dangerous. But iGEM team is very much educated and they nailed it. Of course, there's something like trillions and trillions of microorganisms on your body and inside of your body, really. Most of them are inside of your body. And they outnumber your human cells by 10 to 1. So for every one human cell, you have like 10 other bacteria or microorganisms living in your body. And so, but they're really, really small. And if you weigh 200 pounds, the amount, so if you weigh 200 pounds, 2 to 6 pounds of your weight will be, bacteria. So it's quite a lot of bacteria in your body. We need bacteria in our body in order to function properly. They're healthy and they're good for us. And yeah, so bacteria is good. Yeah. All right, so what is DIY bio or do-it-yourself biology? All right, so here's a little quote and it says, "The era of garage biology is upon us. Want to participate? Take a moment to buy yourself a molecular biology lab on eBay." All right, so what do we see here? We see this guy pipetting stuff. I notice he's not wearing a lab coat, you know, but he's got gloves on and he's got a pipette. So pipettes, so this is for a high school audience, right? So pipettes are instruments that are used to move very small volumes around from one tube to another tube, okay? And they can be fairly accurate, but there's special, you know, tools used for molecular biology. And so he's pipetting something. This thing is an ice block. Let's see if I've got a pointer. There we go. This thing is an ice block, sort of like a cooler that you can put in your freezer, you know, if you're like going camping or something. But it's made for very small tubes. And so it holds temperature, it's cold. And so this guy's pipetting something at a cold temperature. And notice that it's just like in his garage. Like, look at this, like there's like a broken fan, you know, there's some WD-40 back there, all kinds of stuff. I don't, you know, just like, I don't know, it's just in his garage. He's doing a biology experiment. And I have to say, I quite love this. I think it's quite amazing. A few other things he's got in here, pipette tips. This red thing is a thermocycler. It's something that you can put a tube in and raise temperature and hold that temperature, and, you know, for a certain number of time. And this is a centrifuge. So even though he's just doing biology in his garage, you know, he still has specialized equipment, and probably costs him a bit of money to get all these things together, you know. If I was setting up a lab in a garage, I'll probably try to get equipment from a lab that's shutting down or something like that in order to cut down on costs. But also notice the big smile on this person's face. Like, this person is loving this, right? He's just doing biology in his lab. Yeah, open arms, open legs. Not great in my opinion for many reasons we can get into. Okay, so yeah, so people are interested in doing biology at their houses. Okay, so a more traditional setting would be like an academic research lab, and we'll get into that in a second. But you can just do biology at home, which is pretty cool, I think. So where does this DIY bio movement come from? It turns out that it actually traces back to iGEM. All right, so hello iGEM team. I don't know if you guys knew this. Have you guys seen this picture on the iGEM website? I see a couple of you shaking your heads like, yeah. Okay, so these are the iGEM founders, Randy Redberg, Tom Knight, and Drew Endy. This is amazing. This is Drew Endy. So it's a very blurry picture. It's all that I could find. But this is Drew Endy. And the reason I'm showing you this picture, you know, Drew gave a presentation for this group not so long ago, right? And you know what? Drew Endy is actually very influential in the synthetic biology field, and in the DIY bio field, and of course iGEM. And so it's quite amazing that we just had him just give a presentation for this group, right? Drew Endy is an incredible visionary. Okay, so this whole movement was started like in 2004 or something. And so he really has these like big picture ideas about, I mean, if you just talk to him like on any given day, he's talking on a level up here, and he has these big picture things. And that's, I think, something really amazing about Drew, is that he's this incredible visionary. All right, but here's a link, so go read up about it if you want to know more. But this is a great picture. All right, so what does like a more traditional kind of lab look like? And so I took this picture because I didn't want to take a picture from another lab without permission. This is me, believe it or not, that's me. And this is other students in the teaching lab. And we're doing an experiment. So similar to the DIY bio thing, you see that I have a pipette, I have gloves. Let's see, we're doing a Western blot. So we're actually loading this gel, and we've got electrophoresis, equipment. Now this lab is much better equipped than the garage lab we looked at, okay? We have ice bucket, we have a transfer machine. What else is there? If you look back here, you'll see there's a shower. Like, what on earth? There's these fume hoods in the back. They're really nice because let's say you do something that, you work with something that gives off a bad smell or something like that. I don't know if you've worked with beta-mercaptoethanol, any of you guys, but you can put it in this thing and it just sucks air in and out. So instead of toxic fumes or smelly things being in the space, this fume hood is great because it will just take the vapors and and the smell and just sort of suck it right out of the room. So yeah, so this lab is really, really well equipped. Much better than the, and it kind of looks a little bit like a mess, but really it's not. There are multiple stations where you can go to do experiments. You can also see there's a thermocycler here, kind of similar to the garage setup, but a much nicer thermocycler. And you can also notice that people have smiles on their faces because they're doing biology. They're loving what they're doing, okay? What else can I point out here? So here's a bunch of like liquids, and it's in another container. This is called secondary containment. This is a bunch of safety precaution things you do just to be safe in a lab environment. Okay. All right. So what are biosafety levels? I know this is like the part that you're like, do we have to talk about this? This is exciting stuff. What are biosafety levels? Who wants to chime in? Come on, somebody help me out. I'm talking a lot.
I would say biosafety levels help us determine essentially the kind of activities that can go on in a lab based on how dangerous they would be to humans or the environment.
Great. All right. Really good answer. Spot on. So yeah, so there are different biosafety levels, and labs are assigned different biosafety levels to work with certain organisms. Okay. So they go from like low risk to high risk. And if you look at biosafety level 1, these things are not known to cause any like disease or harmful, or, they're not harmful to humans, okay? So, and there are things like yeast, Saccharomyces cerevisiae, E. coli, which, you find in your gut, and other non-infectious bacteria. And so in the teaching lab, and at a DIY bio, so if you set up a lab in your home, you can work with any of these BSL-1 organisms, and that's totally fine. And it turns out that a lot of synthetic biology just uses some regular BSL-1 organisms that's totally safe to use, okay? Now, if you want to work with something a bit more dangerous, so a biosafety level 2 lab will work with something like salmonella, hepatitis. Okay, so they're a bit more dangerous and they can be more harmful to you. And the lab, again, must be equipped to handle these organisms, okay? And then biosafety level 3 is much, much higher. And so there are things like West Nile virus, all kinds of weird stuff, SARS, okay? And then a BSL-4 would be something like Ebola or smallpox, something that has to be contained. You don't want that thing to go out at all. Very, very, very dangerous stuff. Now, because we're in this kind of weird pandemic era, I thought I would just show you where SARS-CoV-2 is located. So SARS-CoV-2 is the name of the virus that causes COVID-19, okay? If you're working in a diagnostic lab, you're just testing to see whether it's present or not, just for diagnostics. It can be done in a biosafety level 2 lab, okay? And these are guidelines from the Center of Disease Control, from the NIH. If you are, however, isolating the virus and you're working with a pure virus, it must be done in a biosafety level 3. So yeah, so this, hopefully this sort of gives you an idea of how dangerous this virus is that is affecting us at this time, okay? All right, so yeah, so just for like doing biology at home, most things are like biosafety level 1, and it's totally safe to work with. I think when people are not educated about biosafety levels and about organisms and how they affect you, then they're afraid, and I think fear causes a lot of irrational thinking or whatever. But I see no reason why DIY bio should not be an awesome movement and can be done very, very, very safely, okay? Yeah, okay, so let's talk briefly about engineered controls, and I'll just do this very briefly so that you don't fall asleep and that, and that we can go on with the conversation. But if you work with something very, very, very dangerous, how do you make sure that it doesn't get out and like cause a lot of harm, okay? And so there are very highly effective ways and then least effective ways to do that. But let's just say we work with like a virus like HIV or something like that, okay? The most effective way is to not work with it at all, apparently, which, if you're like a scientist, doesn't really make sense. If I'm an HIV, I'm studying the virus and I'm looking at HIV-1 or whatever, and I'm working with the virus, then you need to work with the virus, you know, in a lab somewhere. So this is not a good option for a lot of people. In this category would be something like simulation. Can you simulate the experiment, you know, computationally or something like that? In biology, a lot of things are sort of unpredictable, and unfortunately you can't simulate things. You need to sort of test things experimentally. And then you can, you know, there are different levels of control. You can substitute the virus to maybe work with a virus that's less dangerous. And here's this thing called engineering controls. These are things that are built into a lab. Things like a laminar flow hood, you know, an autoclave. So engineered controls, things that are engineered into the lab space to help you safely work with a dangerous organism or dangerous substance. And then administrative controls, again, less effective, but actually very, very important. These are sort of like your operating procedures, okay? Things like, just like training, for example, like tie your hair when there's an open flame, or, you know, just sort of like the way you go about working with a dangerous substance in order to make sure that it's a bit safer. And then sort of the least effective is just PPE, you know, personal protective equipment, okay? But yeah, so engineered controls are kind of important, and there are things that I think lack in a DIY bio setting, and, you know, just because of lack of funding and things like that, you know, money to buy all this expensive equipment or whatever. So here is a sort of a lab, and this is not like a BSL-1. This is probably like a BSL-2, BSL-3 lab. And my apologies for the not so great picture, but I think you guys can see it. Here's a door and there's a sign. It says, hey, this is a BSL-2 or BSL-3 lab. And you cannot enter this lab without an access card, okay? So you get in here and then now there's another door and it looks like there's a shower or something here. You get all your protective gear on and then you go into the lab, okay? And then once you're in the lab, there are very specific sort of standard operating procedures. How do you work with things? Where do you work with it? How many people should be around you, you know? When I worked in the HIV lab, for example, it was like a BSL-3 lab, so it was kind of a high-level type lab. There was a autoclave in the lab with 2 doors, one door on the inside and one door on the outside of the lab. And the reason why this is a very important engineered control is if you have any waste that you generate by, you know, that came in contact with the virus, in fact, any waste, the only way that it can leave this facility is through the autoclave. So you put it in the autoclave, you close the door, it raises the temperature very, very high, pressure very high, kills anything that's in there. And then the only way for it to leave is then on the outside. So you go to using the outside door to take all the trash out. So this is a good example of an engineered control to make sure that things can be contained and not just get out and be sort of dangerous, you know, infect other people and be dangerous for other people. All right. So I've spoken quite a lot about a lot of things. I don't even know how I'm doing on time. I feel like I'm rushing. But yeah, it's only 15 minutes. We're going pretty fast. But so let's compare sort of like a home garage, do-it-yourself biology setup, okay, with more of a traditional lab, okay? And so I think the most important thing that both of these things should be fun, you know, otherwise why are you doing it? You know what I mean? So they are fun. It's really fun to do biology and you can do it wherever. When you set up a lab at your home, you know, you should really stay within a BSL-1, you know, sort of organisms that you're working with. You know, I think that's important. And so it is safe if you are informed about what you're working with, of course, and if you stay within BSL-1 guidelines. The nice thing about it is that it is accessible, meaning you can do it wherever. You can set up a lab in your kitchen, you can set up a lab in your garage. You have limited engineered controls, okay? You know, this garage doesn't have an autoclave. It doesn't have things built into the building that makes it safe for higher level things, okay? And so you have limited engineered controls. And I would say, even though it is accessible, I think, my opinion, personal opinion, is that at-home biology is still kind of expensive, unfortunately. You know, when you buy these reagents and these pipettes and all these things, it is still expensive. And so I think that limits the number of people who can actually use it, which is very unfortunate. And so if we look at a more traditional lab, you know, the nice thing is, so this example, I'm here and I'm showing the student how to do an experiment, but I have like years and years of experience. And so I can show the student, hey, this is how we do things safely. And I think in the teaching lab, and for maybe someone like you who starts out, that is very nice. It's nice to have someone who's knowledgeable, who has prior experience, and you can say, hey, this is how we balance the centrifuge. This is how we dispose of these chemicals. This is how we do things. Why? Because it gives you confidence to not worry so much about doing things wrong, but just to focus on the experiment, because I'm there and I can help you, you know, with safety things. And you can actually learn quite a lot from another person who, you know, has prior experience with things, just by listening and sort of, you know, instead of figuring everything out by yourself, you can learn a lot from having someone else around. A more traditional lab is not very accessible, okay? You have to, for example, if you're on Stanford campus, then you will have to go to a principal investigator, a PI, and say, hey, you have a lab, can I work in your lab? And then oftentimes they will say, you know what, no, the lab is a bit full right now. Or, you know, so it's not very accessible, okay? You can't just do it anywhere. You have to go to academic institution or a medical research institution or something like that in order to get access. Good thing about it, you have built-in engineered controls, okay? But I would say that it is very, very, very expensive. So doing this kind of work is very, very expensive. So let's see, do you guys have anything you want to add comparing DIY bio with traditional labs? Something I didn't mention.
I think that one interesting thing is there's a little bit of a middle place in between, you know, a traditional lab and a DIY bio that the iGEM team has been trying to take advantage of, which is like community labs, which are somewhere in between the two.
Is anybody on the iGEM team working in a community lab?
Yeah, 2 of us are right now.
Awesome. Do you mind if I ask where they are located?
It's at BioCurious in San Jose.
Okay, nice. Are both of you working at BioCurious?
Yeah.
Very, very awesome. So these are labs that sort of a normal person can go to and just sort of go do experiments. So I'm curious, is it expensive to go to this lab, to this community lab?
So it's actually really cheap. It's $100 a month for a membership and a little bit extra for like onboarding, but it's been pretty affordable for both of us so far.
That's amazing. Do they have safety training and things like that?
Yeah, there's like an online safety training. I don't know if they have a, normally not during a pandemic, if they have like a better safety training in person, but it was online. And then they have like community reagents and things, which is really useful.
Wow, that's pretty awesome. Okay. Yeah, so I have no experience with community labs. I know of them, of course. I know there's one in San Francisco as well. That's pretty awesome. Yeah. All right, we'll have some questions for discussion, and then we'll go through a few of these. And then again, when you talk to the high school students, you guys should just run with this and maybe pause it or something. I don't know how you want to do it, but let's go through these. So how many of you have done any microbiology at home? All right. Yes. Very enthusiastic. Yes. What microbiology have you done at home? Have you made kombucha?
No, I've just left cups sitting next to my bed until I have my own microbiology experiment.
That's great. That is awesome. So you've made penicillin or something like that. I don't know. Anybody else? Have you done any microbiology at home? Have you bought a kit maybe? No?
I once bought like a bioluminescent bacteria kit. It was pretty fun.
Nice. I'm sending kits to our students for BioE44. Yeah, because apparently I'm teaching a lab course, but we're not allowed to be in the lab. So this DIY bio is going to be sort of an overarching theme for BioE44. Yeah, in the fall. So that's going to be quite interesting. All right, should everyone be allowed to engage in DIY bio experiments? What do you guys think about this?
I mean, I think I don't know if the question is should, because there's really no way that you can know who's doing it or not. So it'd be like, is it even possible to control? That's my thought.
Yeah, so an interesting point, you know, should everyone be allowed to engage in DIY by experience? What if there's somebody who's got like mental problems and they want to make something dangerous at their house? Like what if they want to make a dangerous virus? Should they be allowed to do that?
I think that perhaps one answer would be not looking to ban people, but looking to ban access to certain things that would enable them to do that.
Okay. That's a good answer. Yeah, because at the end of the day, you can't just control everything that people do, right? Yeah. All right. When is it not safe to work with pathogens at home? Yeah, I should say probably never, because, you know, pathogens, you know, okay. But yeah, you know, so of course, it comes back to the BSL safety level 1 thing, you know, so, you know, yeah, okay. So who's doing the experiments and who's planning the experiments? You know, what kind of checks are available? Like who checks up on people and doing these things? And how much control should, let's say, the government have? Like these are big questions, and I'm assuming that iGEM is talking a lot about ethics and ethical things, okay, with, you know, especially since you're not in the lab. Okay, so what about biosynthetic organisms? Okay, so I'm working with E. coli. You know, I am adding DNA to these bacteria. What if I by accident make a new organism? And so it's a new bacteria, right? It's like I've changed the DNA so much that it's a new sort of bacteria. And then there are questions, you know, so what if this, I'm in my garage making like stuff, and what if I accidentally make some weird biosynthetic organism and it gets released out into like nature, and it sort of like spreads everywhere, you know? Some people, so you were mentioning the, you were mentioning, what did you just mention? So yeah, so some people will like make these bioluminescent plants. I don't know if you've heard about these things, and so they will glow or whatever, and they're pretty awesome. But what if these plants now get outside and they, you know, they're not indigenous, and they, you know, they might, I don't know, what if it's bad for the environment or something like that? All right. All right, I've listed some references for you guys. The top one, I really like this article, and I like this one as well because it puts a positive spin on DIY biology. And so when you talk to high school students, I hope that I would encourage you to put a positive spin on it. I think it's good for people to be, to have access to, you know, all kinds of experiments, molecular biology at home, microbiology at home. And that, you know, I think it can be done safely. And I think just with a bit of sort of you know, creating awareness of how to do it safely, stay within BSL level 1. Don't try to do crazy things. There are all kinds of scary stuff out there, really good examples of people trying to combat herpes virus with CRISPR and injecting themselves with all these things. I purposely did not add that to this presentation because I am aware that we'll be talking to high school students, and you don't just want to scare everybody. But yeah, I want to encourage you to put a positive spin on it. So with that, I am done with my talk. Does anybody have any comments, or anybody want to say anything?
I had a question actually. At the kind of last discussion question, talking about like something that could kind of accidentally be created in a DIY lab. What, say somebody is like a good agent, right? They're not trying to do anything dangerous. They're working with like a BS level 1 organism. What are, if you know, because I personally don't, what are the likelihoods that they would create something like that accidentally?
Yeah, I think it will be very low actually. I think it's not very likely to create, but it can happen, right? So yeah, so I don't know. So what if you accidentally mix? So I'll give an example with viruses. Okay, let's say you have like a viral infection. Let's say you have like HIV or something like that, and then you also get like the flu virus. It is possible for a new virus to form out of these things. So I'm like, I should be cautious here because I don't want to scare everybody, okay? But you can essentially create by accident, your body can create a super virus, which will combine parts of both of these viruses, okay? And those things are not something we can really predict or have control over, okay? So there are rare instances where it will be out of your control and maybe something bad happens and that something is created, okay? But I think if you stick to just like general safety things, like the way you dispose your bacteria, even if you're at home, maybe somebody teaches you how to use bleach and you just, instead of just dumping your bacteria down a sink before you do that, sodium hypochlorite, which is in bleach, will kill everything, okay? And it will also just evaporate. So you can wipe surfaces with it. You can clean up well afterwards. You can bleach your bacteria before you dump it down the sink. And bleaching bacteria and dumping down the sink is actually a safe thing to do. And it's fine to do that even in a BSL-1 lab, but it will kill everything before you dump it out. But yeah, I don't really know how that is, we are able to control something like that, to be honest with you, because there are instances where things like this can happen. Yeah.
I kind of had a question where when you look at something like the field of gene therapy, it only took one mistake to really set the field back, with Jesse Gelsinger. So do you think that one mistake in the DIY biospace will set the field back decades?
That's a really good point. So Jesse Gelsinger, what an awesome example, right? Who had this like ornithine something gene, and so they used a virus to try and add this thing back to his body, and it should have worked just fine. This is in the early 2000s. But what happened was the viral therapy, so viruses deliver DNA, okay? And so they used the virus to deliver this DNA to restore his body function of his, I believe of his, is it his kidneys or his liver? Not 100% sure right now. But make a long story short, the virus load or something wasn't 100% correct. It was tested in monkeys and all these things before. It looked safe and wonderful. But when he got the treatment, his body had this really severe immune response and he ended up dying. And just like you said, it set back the entire gene therapy field for many, 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 many years. I mean, everything was just stopped. The Bush administration stopped a lot of, immediately just stopped a lot of, of gene therapy. Yeah, so I think, yes, if something bad happens, that it will probably set back, synthetic biology, and especially sort of accessible biology for everybody by quite a lot. Let's flip that around for a second, though. What do you think are some awesome things that can happen from synthetic biology? So here we're in this weird pandemic era, so I don't know how much we can actually do to maybe, at your home, like in terms of to like, work on diseases and things like this. But what are some positive things we can do? What if you can create your own medicine? That'll be awesome, right? Especially if it's like an underserved area where healthcare is so expensive, but just because you have the knowledge of how to use these tools correctly, you can do amazing things, right? So what if you can make medicine? What if you can, I don't know, there's a lot of positive things. Can anybody think of something cool we can do with synthetic biology? I mean, besides just creating awesome things, you know, which
the open
is
source
awesome.
insulin project.
There you go.
I mean, there are people who create art with synthetic biology. You know, we've seen like pictures of people using fluorescent proteins and bacteria to create really cool things.
Yeah. I'm all about that. Like I like that a lot. I had 2 students in one of our 44 labs who had this art project, and they just, you know, they use like one sugar and then it turns like red, and another sugar, different kind of sugar, and then the plate will turn like green or something like that. And it's awesome. There's definitely a place for that, and we shouldn't taboo that. I think it's pretty awesome. All right, I think it's probably a good time to stop recording, and then want to encourage you guys to just run with it with the students. And, you know, I think you've had a lot of these type of discussions with iGEM already, if I'm correct. And so, you know, more than those students, so just go with that. And yeah, I got some papers that I listed to give you a bit more, yeah, a bit more information if you want to go with that. So. All right.